So now, if you look at this, I can now look at this bilayer and see that I have a bunch of membranes embedded. They're trying to show me that I have a membrane that's connected, a protein, connected to these glucose molecules, which is part of my glycocalyx. Then they're showing me I have a protein that looks like it's got a channel down through the middle of it. Then I look like I've got a protein that looks like it's got some on the inside, some on the outside. Are you seeing how there's different proteins that are found? So some of my proteins can be attached to this glucose chain. But then right here, I'm seeing this chain existing, and it doesn't look like it's attached to a protein. It's attached to the phosphate of one of these phospholipids. So they are either referred to as being, with these carbohydrate chains, either a glycoprotein, meaning that glucose chain is connected to the protein, or a glycolipid, meaning that that carbohydrate chain is connected to the lipid portion of that bilayer. The reason that these chains are important, their abilities, they can help with the cell communication. If a cell needs to communicate to another cell, they can serve as receptors. For example, hormones moving through the blood that will make it to that extracellular fluid, those chains can act like a receptor to accept that hormone. Those chains can act in the immune system. So that glycocalyx, these glycolipids and the glycoproteins, they are helping protect and the function of the cell. So that glycocalyx that's surrounding it is very important. Because there is so much embedded into this phospholipid bilayer, have you ever, mosaic, are you familiar with the term mosaic? Looks like a bunch of stuff kind of put together. They call it the fluid mosaic model, because having all these proteins, having the bilayer, you're going to see that there's cholesterol in there, having so many molecules embedded into this bilayer makes it look mosaic. And because this is in a fluid environment, it actually looks like, when you're looking at a cell, that all of this stuff just simply floats. The cell's just in there doing its little job, woo-hoo, 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 and all the stuff in the membranes just boom, boom, boom. Wheels on the bus go round and round. I'm so glad all of you smile. Nobody else smiles at me. So, you've got all this stuff, and it literally just looks like it's floating all through the fluid. So, it's termed the fluid mosaic model. Every single part has something it needs to do. Every single part. In the membrane, yes, phospholipids dominate. Cholesterol helps dominate. Now, you hear the term cholesterol. What does it kind of make you think about?
Heart problems.
Heart problems. The stuff that when you go to the doctor and they take blood comes back and your doctor's like, your cholesterol's high, you need to cut that fat out. Okay, not that type of cholesterol, okay? It is a cholesterol molecule, but it's strictly for structure, okay? The little shape of it, and because cholesterol is a hardened molecule, it provides structure to the membrane. The phospholipids, do you happen to remember, because I think we talked about it on Monday, where they come from? Do you remember how we've got those 3 biological molecules, 4 technically, but 3 that we consume? The sugars, the proteins, the lipids. And lipids had the 3 fatty acid tails. Nicole?
Doesn't it like one, like, it starts with a G, I know that. Very fatty acids and, never mind.
You're thinking probably about the glycerol portion
Yes.
of the fatty acid. Well, our body, when we take in the fats and the structure that they have, and the fact that they've got the little 3 fatty acid tails, okay? Remember, the body has to break those down into their component parts so that we end up with 1, 1, 1, okay? So that when the body breaks it down, it'll have this part, but it'll only be 1. But if you get 2 or more together, they do this because of their structure. So they form like that because we have the phosphate head, the lipid tails. Now let's think about this for a second, okay? The heads, the phosphate heads, point towards the water environments. The head here points to my extracellular fluid. This one here to my intracellular fluid. Those lipid tails are in between because that is the fatty material. Fats and water don't mix. So you get 2 or more together, they form this way, and that creates our membranes. Because the phosphate heads are hydrophilic because they love water. The tails, those lipid tails, are hydrophobic. They fear water. And that's a perfect environment that has just gotten created. Like I was saying, the cholesterol is a stiff molecule. But let's think about this for a second. We just said that this membrane is fluid because we are animal cells. If we had a cell wall, oh, okay, it has a cell wall like you see in plants, okay, like that, like the bricks on the wall right there, because that's what plants have to do. And so everything's cellularly stiff. So if we had something that was making our cells really, really stiff, okay, we wouldn't have a lot of fluid movement. Thank you. So what we end up with in this fluid environment, we end up with a cell membrane that, because of the cholesterol that's present, has some structure, but at the same time gets to do this. Literally flatten. If you've emptied your bladder this morning, the cells of your bladder went. That's cool. Okay? Then we have to make sure stuff can move. All right? And that membrane will allow for that. Because of the structure of the plasma membrane, have you guys ever seen, like for example, with in vitro fertilization, and they'll show like the picture of an egg and the needle trying to insert onto the egg? Have you ever seen that? I'll see if I can find one over the break, a video of it. That's the perfect example of how protective this membrane is. You will literally see the egg, which is huge, by the way, okay? Still microscopic though. You'll see the egg and you'll see them coming at it with a needle, and that needle actually pokes in and the membrane pushes in around it. They have to push pretty hard with that needle to put the sperm in for fertilization to take place, because that membrane is so tough. It literally pushes in and pops back out because it wants to protect the inside of that cell. So what'll happen, they push in, but eventually that needle gets to break through. But as soon as that needle gets pulled back out, it automatically arranges back to the double membrane. It didn't like getting penetrated, but it did, and then it literally went right back together. So these membranes, they're pretty tough. And we can see that the membranes can fuse together. For example, once that egg gets fertilized, you've got the one cell that's now been fertilized, it has to become two. So you actually get to see membranes doing this number. So our animal cells are able to do that. So now, if we look at those parts that are present in the plasma membrane, I still want to remind you, proteins work, okay? When you hear the term protein, let it make you think of some type of work that needs to be done, okay? So in this plasma membrane, the proteins that exist, we're going to say they are integral or peripheral. Another term that you might hear, intrinsic or extrinsic. Intrinsic to the inside, extrinsic to the outside. Integral, being a part of, peripheral, to the outside of, okay? So in the proteins that can exist, they can be